So I guess Srinivas told us that 1/3 in this room would perhaps die of cancer. Now that's a little scary. But uh, what if we could sort of have some mechanism whereby we know that we have cancer, where we know that we are predisposed to having cancer. So obviously the answer lies in our genes and most of us know that. But how do you actually find out that? So you have gene editing tools like CRISPR, Cas9, etc. So I've got just the right person to explain what this all mean. So I want to call on stage now Pranav Anam, who is the co-founder of the Gene Box. Please give him a big hand.
Thank you, Leslie, for the kind words. So at the Gene Box, our simple goal is we want to be a data science company that is going to build interventions on top of your genetics and your environmental factors. Because even if you place two identical twins in different environments, they'll be up to 33% different. That is how much a role your environment plays in. However, today I'm here specifically to talk about genome editing, the possibilities, and not just human health, but how we can affect the health of the earth globally as well, and see how this goes forward. So in a nutshell, each one of us, in fact, every cell in our body has around 6.6 GB of data. That's your DNA. It's roughly around 3 billion base pairs. And that's how that's how big the base pairs are. So essentially what we do is there are 4 bases, A, C, T, and G. So they're there, adenine, cytosine, thymine, and guanine. These are the 4 letters which essentially define your entire DNA sequence. This is just a repeat of these 4 letters. Sounds very simple. So now, let me come down to a few interesting mutations that people know about or may have heard. One is superimmunity. I'm sure people here have heard about sickle cell anemia. It's a very dangerous disease to have, but if you only have one of those markers, not 2, then you're actually immune to malaria. That's one of the key things. The other one is super strength. So we all have muscles, right? When we are growing, there is an enzyme which is secreted to stop the growth of the muscles. In some individuals, that enzyme isn't present. So you have like super strong individuals maybe like Hulk. Then you have people with super flexibility, contortionist artists, they can bend the wrist backwards. You have people with super dense bones. So there is a family in the US. This guy at the age of almost 70 plus had a major car accident. The car was totaled, but this guy walked away without a broken finger. And when they researched more on him, they realized in 5 generations in his family, nobody had a broken bone. Turns out one gene gives them extremely dense bones, almost to the strength of adamantium, that is Wolverine, essentially. And the downside of that is you're so heavy that these people mostly can't swim. They sink very fast because the density of the bones is very, very high. Believe it or not, there is a 1% audience, 1% population in North America, Northern Europe, which is specifically immune to AIDS. These people can never get HIV. That's how different we humans can be. Then the next is tetrachromatic vision. Essentially, you can see 4 times the color of a normal human being. Mostly females have this mutation. So sometimes when we men are wondering, yeah, it's a pink, but not which pink, you'll probably understand why. There's another small tribe in one of the islands which has crystal clear underwater vision. So they can see as clearly as you can see now, but that is underwater. And then there are certain individuals who are built for high altitude, essentially. Now, the reason I'm discussing all of this is towards the end of my presentation, I'll come back to these. So what is the buzzword about genome editing? So it's nothing but a simple process to basically edit the base pairs that we just spoke about, the AGCT, and probably change the sequence. Now, we've been attempting to do a lot of these things for a few years now. Primarily, that's what one of the starting this is something called as an engineered nuclease, which is also called as a vector, which was used to edit the genome. So what does it happen, right? What does it really, really mean in a nutshell? It is an efficient copy-paste system, right? So the first version that we created could not cut at a particular location, but close to it, and it could not upload as much data into the DNA as we wanted. However, with CRISPR, it is as accurate as it can be. It is a very low-tech module. Believe it or not, the Cas9 module is actually from a bacteria. It helps defend itself from viruses infecting its DNA. So that's how Cas9 is, and CRISPR is your tool, basically your molecular scissor. So going forward, I'll tell you how this is useful in various applications. As just discussed, this is the slide. So what is genome editing used for today? So one of the key things that we are using it today for is agriculture, therapeutic potential, a lot of research, and possibly affecting evolution. So let's look at the therapeutic potential. Now, cystic fibrosis, it's something as we call as a monogenetic disease, which means that only one gene is responsible for this disease. What happens in this disease particularly is there isn't an enzyme which is secreted, because of which there's a mucus formation, and there's a high mortality rate in children. So what we could do theoretically in this case is we could take the patient's stem cell, use CRISPR-Cas9, add in a new gene, which is going to make that protein, that molecule that is needed to prevent excess mucus formation in the lungs, okay? And inject that patient with these new stem cells, make sure they get engrafted, and that person has a chance to survive and live without cystic fibrosis. This is pretty cool because you don't have to do it in the entire body, because there's only an enzyme which is not being manufactured. You want to replace that. So you're adding just another insert into the body, which is going to make a new molecule. So for those of you who don't know, the DNA is actually a manual for every possible molecule that is made in your body. So all your enzymes, all your proteins, all your receptors are coded through your DNA. And this is something I hope comes into practice very soon, because it is a life-saving tool. China has completed the first trial using CRISPR-Cas9. This was done for lung cancer patients. That's all the data I have on it right now. There's not been a publication on it, but we really know about the study because it's being tracked through a research platform. Now, something which is very interesting that really, really excites me about the CRISPR thing is we could potentially bring back the woolly mammoth. And it sounds fun and fancy, but why is it important? So I'll give you a simple example. How many of you here, I'm sure not many, but would know that there is the introduction of wolves in the Yosemite National Park has totally changed the ecosystem there. It has stopped the soil erosion, the rivers are not changing course anymore, and the ecosystem which was up in a mess, it's now all sorted. So what happened? So what used to happen is, because there were no predators in the park, the herbivores were grazing everything everywhere along. And that was not giving the soil the firmness. There were no roots essentially binding the soil, and there was soil erosion, which led to the river banks expanding, changing the entire ecosystem. When the wolves were brought in, what happened is they started restricting the herbivores to a particular location. So the grazing fields became narrower. That made the soil more firmer, and over a period of time, you could see the results coming back. Now, the elephants today are the most endangered species that we know about. But at the same time, these are very important to the ecosystem of the earth. Especially now, because of the global warming, what's happening is the permafrost, which is basically a layer below your soil, near the Arctic regions, is actually melting. It has a lot of greenhouse gas effects. It's not really helpful for us. So what is happening is, if we are theoretically possibly do this, the hybrid woolly mammoth 2.0 would be shifted there, along with other new elephants or the hybrid elephants. Their walking would puncture the perma soil, interacted with the cool air, preventing from melting. And in the summer, what would happen is these woolly mammoths would actually bring down trees. So essentially, that will help grass grow and firm the soil and use the nutrients better, hence preventing global warming. Now, the woolly mammoth actually went extinct 4,500 years ago, roughly, purely because of the end of the Ice Age, and also because of human hunting. But why specifically look at this mammoth alone, is the next question. Can we bring back tigers which are extinct now? Can we bring back other species that are getting extinct? That is a possibility. So what is it that scientists are trying to do in this case? They've been able to isolate the DNA from a fossilized woolly mammoth. And now we're looking at genes specifically towards anti-freezing blood, that thick layer of wool, large ears, the very traits that help the woolly mammoth survive the Ice Age, and then try and put them in the northern region. Similarly, we're doing this for agriculture as well. We're trying to bring in sometimes more vitamins, more minerals into a fruit. We're always on the look for making new compounds synthetically, but we can also grow them in a plant. Plant biotechnology, believe it or not, has had a very wide application. In fact, it is used to clear up heavy metal land pollutions. So essentially, you would put in a gene that would help absorb a lot of heavy metals from the land and store it in a particular part of the plant. And that's how eventually over a cycle of maybe 10, 15 years, you could reduce the amount of heavy metal pollutants in a particular piece of land. So what is Holy Grail? Is CRISPR-Cas9 is really there to solve our problems? Initially, I think there's a lot of ethical debate on it, primarily because the uses for it are unbelievable. You could change so many things. You can make crop-resistant, drug-resistant crops or pesticide-resistant crops. You can use it to bring back extinct species. But does it stop there? Is it going to go towards developing advanced humans? Is it going to go towards designer babies? Is it going to go towards giving people adamantium bone strength and Hulk-like muscles? Is it going towards advanced warfare? And this is where the basic criteria for ethics is coming in, and that's why the use of CRISPR needs to be extremely, extremely well regulated. Now the problem is the technology is so cheap that it's very, very difficult to regulate it, and it's not that not a lot of people can capture it and do about it. My goal here is to open you to possibilities on what can be done with genome editing. It's the next way forward. As of now, even today at the GeneBox, we can tell you what you're going to be deficient towards, how your nutrition panel is going to be. We can tell you what medicines you can take. We can tell you what medicines are good for you and not good for you. We can help you adjust your dosages. And we are not even scratching the surface as of now. So also the new advances in genome sequencing. So till Jan this year, if you wanted to buy a genome sequencer, it would cost you a cool $10 million and probably $1,000 as a raw material cost to sequence a genome. But nowadays, the cost has come down for a cool sequencer to less than $1 million. And the sequencers are now tabletop. You can sequence an entire human genome in less than an hour. And the reagent cost is down to $100. But before it hit the market, it's going to be another couple of years. So it will be interesting to see once we have more data, more genes, more different individuals with interesting mutations, how does it get all passed on? That's the way going forward. Thank you for your attention. In the interest of time, that's where I conclude. Thank you.
Thank you, Pranav.